Welcome to EPG Pathshala. I am Aparna, working as scientist in MFPI Food Quality Control Laboratory, Professor Jaishankar, Telangana State Agricultural University, Hyderabad. Today, we will learn about the topic heat preservation and processing. As you all know that heat preservation and processing has been applied since ages for preserving and extending the shelf life of many kinds of food products. Today we will learn about the various types of heat preservation techniques. We will categorize them broadly into three categories and try to understand each category in detail. As we all know that the safety and storage life of many perishable foods can be enhanced by use of high temperatures to inactivate undesirable, disease-causing and spoilage microorganisms, and also to inactivate the enzymes which can lead to food spoilage. So there are three categories of heat treatment and heat preservation. The three categories of thermal preservation are blanching, pasteurization and commercial sterilization. Blanching, as you all know, is the most commonly utilized preservation technique. Blanching is one kind of thermal processing which is applied to many vegetables and some kinds of fruits by exposing them to either heated water or boiling water or steam for a short period of time. Blanching is a food processing operation which is designed specifically to inactivate the enzymes in the plant tissue so that the enzymatic degradation does not occur in the intervals between packaging and thermal processing or during the frozen storage or in the early stages of food dehydration and also after reconstitution of the dehydrated plant foods. It is also done to wilt the vegetable products so as to enable the packaging of the products into containers so that the proper fill weights can be achieved. That is, you can reduce a mass and volume of fruit or vegetable by blanching and reduce the volume and amount of the fruit. It is also done to drive off the inter and intercellular oxygen and other gases which are present in the plant tissues so that the containers will not be deformed by the release of the gases which are naturally present in the fruit and vegetable tissues. Apart from that, they will also make sure that the high internal pressures will not expand the gases within the container and will also permit the formation of a vacuum in the container after the thermal processing is done. So blanching is carried out as a pre-treatment for many kinds of fruit and vegetable processing either before dehydration or canning or any other preservation technique. After understanding the first heat treatment, that is blanching, let us now move forward to the second type of heat processing and preservation, that is pasteurization. Pasteurization is a very extensively used technology of heat preservation which is applied to many categories of foods. Pasteurization is a thermal process that involves using temperatures of at least 72 degrees centigrade for 15 seconds. It is also termed as high temperature short time pasteurization, or it is also called as HTST process. This kind of pasteurization of subjecting the food to at least 72 degrees centigrade for 15 seconds will be done prior to packaging of the food material. Acid foods which have got a pH of less than 4.6 are mainly pasteurized to inactivate the spoilage which can be caused due to microorganisms. Then we also have some pathogenic microorganisms which cannot grow and do not survive very well in acid foods, for example the citrus juices or apple juices, etc. In low-acid food products like milk, etc., the basis for preservation by pasteurization is to inactivate the pathogenic or the disease-causing bacteria and viruses. Many spoilage-causing microorganisms can still survive typical pasteurization process conditions. For example, in milk, the proteolytic and lipolytic bacteria are more heat resistant and can survive the entire pasteurization process. This explains why the typical spoilage pattern of pasteurized milk reflects the proteolytic and lipolytic action of the psychrotrophic and spoilage causing bacteria. The proteolytic changes are nothing but protein degradation in the milk or any other matrix. Similarly, lipolytic changes are the lipid degradation and lipid deterioration associated with the lipids present in the food. Now, because the pasteurization does not kill all the psychotrophic spoilage bacteria in the milk, the pasteurized milk always should be refrigerated to maintain the shelf life of the quality. The durable life date on the milk containers, it will reflect the storage life that can be expected when the milk is stored under refrigeration or at temperatures lower than 4 degrees centigrade. Then after pasteurization, we will try to understand commercial sterilization. Commercial sterilization is a preservation method, and the basis for preservation by commercial sterilization is to destroy both the spoilage as well as disease-causing microorganisms in low-acid foods and acid foods also, thus rendering the food to be commercially sterile. Commercially sterile means the condition obtained in any food that has been processed by the application of heat alone or in combination with other treatment to render the food free from any viable forms of microorganisms, which includes spores which are capable of growing in the food at temperatures at which the food is designed normally to be held during distribution, handling and storage. Therefore, commercial sterilization involves destruction of spoilage-causing bacteria and microorganisms as well as disease-causing microorganisms. Commercially, sterile foods may contain small numbers of thermophilic bacteria spores. However, the spores cannot germinate and produce actively growing cells at the room temperatures. After understanding pasteurization and commercial sterilization, let us now try to understand the next heat preservation technique, that is canning. Canning can be traced back to early 1800s when it was used during the World War II for preservation of foods for a longer period of time for the warriors who were fighting in the wars as well as seamen. Canning involves heating the food with a minimum treatment of at least 121 degrees centigrade moist heat for at least 15 minutes. The process usually involves pre-sealing the food in containers prior to heating and canning. Today, if a canned food is being sterilized, each food particle must receive the heat treatment. Today, if we want to know about the canned food and how it is being sterilized, we have to assume that each food particle must have received the treatment, heat treatment of 121 degrees centigrade for at least 15 minutes. When food is placed in a can, the heat treatment will change since heat transfer to the food takes place at a slower rate. Depending on the size of the can, the time to achieve the sterility can be anywhere between few minutes to several hours. Most commercially sterile products have a shelf life of at least 2 years or more than 2 years if stored under ambient conditions. After understanding canning, let us now try to understand the next heat preservation technique, that is UHT processing, ultra high temperature processing, and we will also understand aseptic packaging. The combination of ultra high temperature processing and aseptic packaging is another form of commercial sterilization. It involves the application of ultra high temperature to the food before packaging and then filling the food into pre-sterilized containers in sterile atmosphere. This process will render the food shelf life and shelf stability and also commercial sterility without the need for refrigeration by storing the food under ambient conditions. Ultra-high temperature processing and aseptic packaging is a relatively new development where the food will be heated to 140 to 150 degrees centigrade very rapidly by direct injection of steam held at a temperature for a short period of 4 to 6 seconds and then immediately cooling in a vacuum chamber to flash off the water added in the form of condensed steam. This is carried out as a continuous flow operation. The decrease in processing time due to higher temperature and the minimal come-up time and the cool-down time leads to a higher quality product. The ultra-high temperature processed food is packaged aseptically into pre-sterilized containers. These are usually cartons which are made from laminated plastic, aluminium and paper which are chemically sterilized with a combination of hydrogen peroxide and heat and then filled in the same piece of equipment which is housed in the sterile environment. The entire process right from processing until packaging takes place in aseptic condition only. There are other forms of packaging that can also be used in aseptic ultra-high temperature processing, such as plastic cans, flexible pouches, thermoformed plastic containers, bag-in-box and bulk totes. Ultra-high temperature aseptically packaged products have a shelf life of 6 months or more than that without refrigeration. It depends on the type of the packaging which is being used for packing the food material. For example, we have the Tetra Packs which are available in the market. The Tetra Pak cartons can eventually be more prone to perforations in the packaging layers, whereas the newer plastic bottles are more resistant to pinhole formation, allowing them to have longer shelf life even than the Tetra Packs. Let us see some examples of food products which are processed with ultra-high temperature processing and aseptic packaging. We have the liquid products like milk, juices, cream, yogurt, wine, salad dressings, etc. And apart from that, we also have some semi-liquid and solid products like baby foods, tomato products, fruit and vegetable juices, soups, etc. which are processed with the ultra-high temperature and aseptic packaging combination. Contrary to the popular opinion, ultra-high temperature processed milk and juices do not contain any added agents to provide the longer shelf life or storage at ambient temperature in the laminated cartons. The products are preserved exclusively through application of heat. It is critical that the sterilized products are transferred into the packaging equipment under aseptic conditions only to avoid contamination after thermal processing. If a product is mentioned that it is UHT treated, it has to be taken into account that the UHT treated products are not aseptically packaged. This gives them the advantage of a longer shelf life at refrigeration temperatures only compared to the conventional pasteurized products. However, this does not produce a shelf-stable product at ambient temperatures due to the possibility of post-processing recontamination, which could be because even if ultra-high treated product is not aseptically packaged, then the post-processing contamination can be quite possible. Now, after understanding the various kinds of heat treatments, we also have to understand how to select the heat treatment for a particular kind of the food. So, let us now try to understand the selection of heat treatments. The intensity of heat treatment employed for a particular food preservation application, it depends upon a number of various factors. The main considerations in selecting the required temperature and time conditions for thermal processing are: what is actually the objective or purpose, whether it is just blanching or pasteurization or to get a commercially sterile product. Then we also need to understand if there are any additional preservation and processing steps, or whether it is just a combined preservation technique method. Then we also need to understand what are the physical and chemical properties of the food and the type of the food for which the heat treatment has to be given. We also need to understand what is the heat resistance of microorganisms which is present in particular food. After understanding the objective, purpose, any additional preservative steps required, the physical and chemical properties of the food and the kind of heat resistance in the microorganisms which are present in the food, we have to proceed with selection of the type and application of heat preservation method. Let us try to understand this by some examples. Foods that will be consumed within a short period of time after processing can have storage life extended by a combination of just simple pasteurization and refrigerated storage. For example, the pasteurized milk, which will be pasteurized, vacuum packaged, and will be consumed by the consumers. Similarly, cured meat also. Then if we want longer storage times at ambient temperature in evacuated sealed containers, it requires the use of commercial sterilization. Then if the time and temperature combination required for pasteurization and commercial sterilization is determined by the most heat resistant, disease causing and spoilage causing microorganisms in a particular food commodity, then we will have to select a combination of heat preservation techniques. For a particular kind of food commodity, the type of thermal processing operation and the rate of heat penetration into the slowest heating portion of the food within a particular container are always governed by the food's physical property. Whether the food is a solid food or a liquid food, or some solid particles are suspended in a liquid food. It is also essential to understand the chemical properties of the food. That is, the pH, the fat content, the presence or absence of any heat-inducible thickening agents, and the food components that have protective or antagonic effects on the thermal resistance of microorganisms. So after understanding all these physical, chemical and other parameters, it is imperative that thermal processing and preservation process should be designed so that the slowest heating portion of the food commodity also receives the specified time temperature thermal treatment to minimize the risk of illness or post-processing spoilage. The thermal processes applied to the foods are governed by heat resistance of the microorganisms in the food. In low acid foods, which are to be thermally processed and vacuum sealed within the gas tight containers, the microorganisms of most concern is Clostridium botulinum. The habitat of Clostridium botulinum can be soil, that is through agricultural or forest produce which enters into the food. Then it could also be through water, whether we are using fresh water or brackish water or marine water. Another source of the Clostridium botulinum contamination could be through the mud, whether it is freshwater mud or saltwater mud. So, the habitat of Clostridium botulinum, which can be from soil or water or mud, has to be taken into consideration. As a consequence, all the foods of agricultural and fisheries origin must be considered as being potentially contaminated with Clostridium botulinum spores. Thermal preservation treatments of low acid foods that are to be packaged and stored under anaerobic conditions must be designed to ensure the destruction of any Clostridium botulinum spores that may be present in the food with the provision of a wide margin of safety. Actively growing vegetative Clostridium botulinum cells produce a very potent neurotoxin. Therefore, it is important to understand the destruction of Clostridium botulinum spores. To determine the thermal resistance of heat-resistant spores in the foods, that is, we have to conduct some inoculated pack studies by using the non-pathogenic spore-forming bacterium. Now let us try to determine the appropriate temperature and time needed to make sure that heat-resistant spores like Clostridium botulinum also are destroyed during the heat processing. For this, we need to understand the thermal death curves. The thermal death curves can be calculated for any kind of microorganism. Let us try to understand what are these thermal death curves. When microorganisms, it can be bacteria such as vegetative cells or spores or molds or viruses and yeast. When these microorganisms are exposed to high temperatures capable of causing death of the organisms, one observes that the population is not killed instantaneously. We generally see that the microbial death during thermal processing follows a logarithmic order. This means that bacteria are killed by heat at a rate that is nearly proportional to the number of microorganisms which are present in the system and in the system which is being heated. For this, we need to have an understanding of the survivor curve or thermal death rate curve. And the survival curve or the thermal death rate curve depicts the logarithmic order of death of the microorganisms. We have to note the time taken to traverse one logarithmic cycle, which represents the time at a constant temperature which is required to kill 90% of the microbial population. The exposure of microorganisms to a specific temperature for a period of time which is required to kill 90% of the microbial population is defined as the decimal reduction time or D value. While the thermal death curve describes the rate of death of a particular microorganism under a specified set of conditions, a thermal death curve can be constructed from a number of thermal death curves by exposing the microorganisms to a variety of temperatures and by determining the decimal reduction time or the D value at each temperature. The thermal death time curve provides information about the time required to kill a particular microorganism in a particular food at a variety of temperatures. All of the time and temperature combinations along the plotted thermal death time curve represent the same killing power with lower temperatures requiring longer time of exposure. One should also note that at any point above the line, that is, for example, 100 minutes at 100 degrees centigrade will ensure that microorganism is killed, while time temperature combinations that fall below the plotted line, that is 100 minutes at 104 degrees centigrade, represent conditions that will not kill all the microorganisms which are present in the food matrix. The thermal death time curve that vegetative bacteria cells have a much lower heat resistance than the spores. The spores will have a thermal death time curve with a higher temperature and set of conditions. Foods that are a combination of solids and liquid components heat up by a combination of convection and conduction heating. Let us first try to see the conduction heating. The location of cold points in relation to the conduction heating have to be taken into consideration in the geometric center of a cylindrical container. Whereas the convection heating has to take into consideration 1/3 of the way up the center axis, which is measured from the bottom of the cylindrical container. By understanding the conduction and convection heating, we have a different kind of combinations which can be set for heat preservation, which can be applicable for different kinds of food matrices. The cold point of a container of the food must receive the required amount of thermal energy to ensure killing of Clostridium botulinum spores or any other spores or microorganisms that might be present to ensure the sufficient margin, safety and quality of the food material. If a food formulation is changed such that the mechanism of heat transfer is altered from convection heating to conduction heating, and if the processing times are not altered to accommodate the change in mechanism of heat transfer, the food could be under processed and could also pose a potential health hazard with respect to botulinism or any other condition. It is important to note that Clostridium botulinum contamination is not very rampant in India. It has not been observed in any kinds of foods, the contamination of Clostridium botulinum. However, problems like E. coli, Salmonella, etc. kind of contaminants are widely prevalent in Indian processed foods. Therefore, it is important to understand the thermal death time curves, etc. for not only Clostridium botulinum, which is not very prevalent in Indian conditions, but it is also important to understand for E. coli and other types of microorganisms where the contamination is high in the Indian foods. So in today's class, we have tried to understand about the various methods of heat processing methods. Under the various methods of heat processing methods, we have tried to understand blanching, pasteurization and sterilization techniques. For more information on this topic, you can refer to any food technology textbooks or textbooks which are related to principles of food preservation. You can also find more information in depth about this topic on heat processing techniques and heat preservation methods which are available. Thank you.